I want to talk a little bit about good scientific practice in MEEG research. As you know, this week we're focusing on the data analysis part of MEEG project. Now, before we move on, I would like to zoom out a little bit and take a larger perspective. For the science that we do to be reliable, we're using a set of rules, implicit and explicit, that we call good scientific practice, that allows us to trust the results we generate, right? These rules define what we consider reliable enough to rely on and build upon. In the MEEG and EEG field, the first good scientific practice guidelines were created in 1977 after an international symposium on cerebral cortex evoked in man, held in Brussels in '74. This is the paper that started, that made the very first recommendations, like disclosing the number, age, sex, handedness of the subjects, describing the apparatus in detail, and defining the nomenclature for various components and so on. Another very influential paper that appeared then later in 2000 by Picton and co-workers started to describe in detail what should be reported and how. But note that these two first papers were about evoked potentials, so nothing about spectral analysis nor about MEEG research. For MEEG, the paper by Joachim Gross et al. In 2013 is a very influential paper. It includes also recommendations for spectral and source estimation analysis. These papers are still very much current, right? And finally, the latest recommendations were made at the OHBM committees and published in Nature Neuroscience like 2 years ago, in 2020. And 2020 is also the year that we at Cutting EEG ran an online conference called Live MEEG on good scientific practices. The S sometimes pops out, sometimes stays in. We did not really have the ambition to create a committee and reflect on creating guidelines, but instead we took a different route. We explored both the regular aspects of current practices in conducting an MEEG project, talking about data collection, data analysis, processing, sharing, publication, and so on, but also things related to other aspects of scientific practice, considering the pitfalls to avoid while preparing a project, the potential problems in the modern ways. Pardon, I'm trying to figure out what this sentence says, sorry. We tried also to talk a little beyond data analysis, exploring what these types of recommendations offer in terms of reflection also on the responsibility of scientists while running MEEG experiments. In today's talk, I will try to get you thinking a little beyond data analysis, so as I just alluded to, and consider things to keep in mind while preparing and running MEEG projects. And if I get the time, I'll reflect a little further about the responsibility of neuroscientists, but I don't think I will have time to do that today. But the paper that you see here was published after Live MEEG, and some of the authors are here. And most of the talk that I'm going to give today is actually relating parts of this paper. And we also edited, as Anne-Sophie mentioned already, this special issue, Advances in Scientific Practice for MEEG Research, which has 26 papers in it, and lots of really good ideas and recommendations. Okay, so what I'm going to talk about today, and I should be finished in 3 minutes normally. That's going to be tough. Today, I'm going to talk about the topics that hinder our scientific inquiry in MEEG. This applies to all the fields of experimental science, but we're going to talk about MEEG in particular. First, I will briefly overview our brain's search for efficiency and how it leads us to conducting biased reasoning in experiments. I will then talk about the so-called questionable research practices and how these intuitive, formerly widely spread practices affect the reliability of our research. Both of these two problems hinder the generalizability of experimental results and pose a major challenge for science in general. And in recent years, the scientific community has developed a number of ways to overcome these problems. And this is what I'm going to mention then to finish. Okay. Now, if you haven't studied psychology or neuroscience, although I know many of you have now, you know that our brains, even if you haven't, you know that our brains are limited in their capacity. But you also probably know that our brains are very efficient and that we use shortcuts to help us overcome these limitations. These shortcuts are called heuristics, and they work most of the time. Faced with a new situation, we usually don't need to analyze in depth this situation before reaching a decision. We use our experience and trust our intuition to come to the right decision most of the time. But in some situations, these heuristics end up leading us astray and reach false or biased conclusions. Heuristics create biases at the perceptual, cognitive, and social level. And I'm going to develop that a little bit on this image here. It is a heuristic of your brain that lets you infer a man or a god's shape in this wave that actually never existed. This image was called Poseidon coming out of the sea. These hallucinations are called pareidolia. This is about the tendency of humans and probably other animals to perceive meaning in meaningless signals, in clouds, in stars, auditory noises. Now, this is a rather dramatic case, obviously. But what happens if you're desperately looking for something significant in your time frequency data? If you're looking at dozens or hundreds of such time frequency maps, running an exploratory analysis on your paradigm, are you sure that you will always be as objective as you think you are? Fortunately, some of us here were trained in experimental psychology and know these limits quite well, and that we have procedures and tools to minimize the chances of falsely seeing something when there is only noise. But it is useful to keep in mind that we can easily be misled by our perceptual system. Now, let me show you a few more of those perceptual biases. And I will focus in particular on color because it is very easily misled. And yes, the effects are quite powerful there. I want to talk about the infamous rainbow color map, also known as jet color map in MATLAB. One may be tempted to say, okay, yeah, so it is used a lot still nowadays in EEG and MEEG research. You see here this time frequency plot. You may be tempted to say very different things. This is the same, same data that generated these maps. So based on the color map, you may be very tempted to say very different things. Look at how the jet color map perceptually amplifies this very small blob here that you don't see in any of the other color maps. This is due to this yellow stripe here that is particularly luminant. You see, this is the luminance as a function of the color here for each of these color maps. And you see this one is very biased. Still, it's the one that is mostly used in many papers. It's decreasing, but still very much in use. Also, it's important to think of the color blindness of people. About 5% of us are colorblind in some way, so they may perceive something very different, and a color map that is well tuned to account for this is important. The Viridis color map, for example, is particularly good for that. The red-blue contrast is also something that is mostly preserved even in colorblind people. So it's also a good way of using it. Okay, the use of color in data visualization is a fascinating area that is a flourishing field of data science. I've noted here a very thorough blog paper on the topic that I used sometimes for designing data representations. It highlights, for instance, the difference or the distinction between sequential, diverging, classed or unclassed, so in blocks, or categorical color maps that are always good to consider when plotting data. Okay, I really recommend this blog. Now, a few words about cognitive biases. There are dozens of ways in which our intuition and gut feelings can lead us astray. And I want to start with a little example here. Visit wood clap again, and I will try to make it smoother this way. Okay, so this was this, and switch to the next question, probably with this point. Okay, now in this list, in the list of numbers that is shown here, 3, 18, 9, 6, 603, and 81. This is an unordered list. One of your, well, there's a rule behind this. I'm sorry, I'm misexplaining this. There's a rule behind these numbers. And I want you to make a hypothesis as to what rule it is, and to test your hypothesis to try to know which, what is this rule, okay? You still have 22 seconds. Please enter everything you can here. So you should test either with 15, 7, or 732. 732 being a number that can be divided by 3 for just, I'm just saying. Okay, and can I reset this before it ends? 3, 2, 1. Okay. Okay. All right. And 62 out of 166 people here. Please hurry up. Don't worry, nobody is going to judge you because you entered the wrong answer. And actually, you know, I mean, I think we are more or less stabilized here. And I'm happy because it works. It's a rather nice example, illustration of what is called the confirmation bias. Our tendency to prioritize confirming our hypothesis over disqualifying them. In this case, the rule could have been that all these numbers are natural numbers. No, this sentence is old. You also that this list of elements are multiples of 3, right? And you sought to confirm your hypothesis rather than looking for some new information like, does a number that is not a multiple of 3 work? Instead, you try to confirm your hypothesis. Now, do you think that this is a good way of proceeding when preparing an experiment, only to confirm what you already know? I'm asking actually. All right, so basically here the idea, I think you got it, right? Testing this number, whether this number works or not in the, fits or not in this list, would have been most informative because you already know that the other numbers work, right? Let me move on quicker. Okay, another, what? No, pardon, that's not my presentation. Oh la la, Matt, this morning. Okay, okay, another example. The confirmation, the sunk cost fallacy. This is our tendency to factor in previous costs while making a decision. When you're working on your project, you may be tempted to take into account the time that you've spent on your data analysis and think like, okay, it's almost there. I'm almost getting this significant effect. I'm not going to give up now. I've been working on this for months. When in fact, if you had been presented with the evidence at once, okay, this test doesn't work, would you keep going? This is the point I want to make. Okay, so cognitive biases are relevant in many fields of human activities. research, but also politics, business, law, and there are really, this is a very relevant field. This graph that I'm sure you cannot see, obviously, comes from this blog post again. You will get my slides, by the way, if you want to look into this. This is a very nice article on cognitive biases. Okay, and a few words now on social biases, and perhaps actually I will have to end the presentation then rather abruptly, I'm afraid. Okay, to finish this very brief overview of biases that can affect the research process, I want to mention social biases. These are biases that occur because of the way we situate ourselves as researchers in a community and in the society in general. They affect the way we think of our work and of that of others, and they can influence what research gets conducted, published, and funded. The majority of studies in MEEG research and in experimental psychology at large are run by so-called weird people, Western educated, industrialized, rich, and democratic people. And the subjects of the experiments are also of the same background. It is a very well-known phenomenon in social psychology that people tend to perceive themselves and others as in-group and out-group and are biased in their reasoning about it. The in-group bias makes you favor a hypothesis of your own group, and the out-group homogeneity bias, for instance, makes you consider people from the other group as more similar to each other than they actually are. So this creates the risk here that weird researchers consider non-weird research as less worth their attention, and that the results produced don't actually generalize to the general population. Social boundaries are hard to break due to these facts. And yes, I want to make also a case about disparity in gender representation. It is still very strong in neuroscience and in all fields. Women are silenced in science and for many reasons. And although measures are taken to increase gender equity, biases remain. However, the more diverse teams in terms of gender have been shown to outperform homogeneous groups in innovation, flexibility, problem solving, and decision making. So to convince you if it needs to be, the imbalance here is shown visually between first authorship between men and women. You see here it's mostly balanced, but for last authorship, you see that men are much more often last authors, prize awardees, seminar speakers, and conference speakers. Solutions have been proposed to this at the individual, group, and institutional level. And I invite you to view a replay of Anaïs Llorenz's talk at Cutting EEG last year on the topic, or even talk to her. She is here in the room on the first row, to talk to her or even read her paper from last year. I'll be really quick on questionable research practices and yes. Okay, please go to WooClap again and change the code to EBCCZF. It's written here. I mean, you have the QR code here, and voila. It's going to be like a little fun game to finish, and you'll be able to come back to me later and talk more if you want to hear a bit how to counter these bad research practices. So, of course, you're waiting for me now to start this thing, right? This slide, no, present of view, no. This is it here, no. Where is it? Okay, up, it's here. Let me bring that back to myself. Where is it? I'll be right back. Up. Up. And exit this. Sorry, I had to. Well, actually, you see the question, don't you? No, you don't? Okay, so that's good. Okay, now you see it. Voilà. Okay, so say you've designed your experiment perfectly. It's really well thought. You've done power analysis, everything you need to do to conduct really good research. Still, you don't get the significant effect you're after. What do? Do you archive it, reflect, and start over again? Non-significant results are undecided, and you need to decide, right? You call it a pilot experiment. You change one parameter and you run a new experiment. Or you try to publish it anyway, as originally planned. The world needs to know about it, about your failure. Or you try a different channel selection. You change your filter. You change your ICA cleaning strategy. There has to be some difference between these conditions, right? Or you add more data to help revealing a weak effect. You invite a bunch more subjects. Okay, all right, hurry up. I'm showing you the results. It's actually quite encouraging. You don't see it, right? You don't see the results, right? I only, okay. Allez, you're almost done, but if you haven't voted yet, do it now in the 5, 4, 3, 2, 1 second. Okay, because after that, you may be biased by this result, which is quite encouraging. I'm glad to say that most of you have answered correctly. Indeed, you need to publish it as it was planned originally, because if you start reflecting and changing your plan on the fly, it's going to lead you astray. And let me find my my slides. Where are they? Okay, puff. There we have it. Did you take a picture? Okay. So just a few words about the three typical effects that I'm talking about here. The first questionable practice, discarding the studies that don't work, either by just burying them or by calling it pilot and starting over again, leads to the so-called file drawer effect. So you just stuff your study in the file drawer and you just move on. This creates a potential waste of time for those people who are coming with a well-designed experiment of the same kind afterwards and will try it over and over without knowing that it doesn't work. It also happens at a larger scale that if we all publish only significant results, and this is what has happened for a long time, in the long run, it creates an overestimation of the effect size. If it's not exactly clear why it does so right now, come to talk to me after, because I will have to skip the slide that explains it. Second questionable research practice happens when you tweak your analysis or you massage your data. It's also called p-hacking. It's very easy to do. Admit it, you've done it. I do. I did. It's just changing some parameters. You know, the channel you use, you remove those parietal channels that don't make any sense anyway, or you change the filter. There's a lot of high frequency noise here, so let me lower my low pass a little bit until the expected effects reaches significance. Bingo, you have it, you write your paper, you submit, you revise, you publish. And it's a fairly natural thing to do, especially when you're sort of panicking because of the lack of time like I am experiencing right now. Finally, the third questionable research practice is called optional stopping. It happens when you decide whether or not to collect some more data after looking at it. If you do this, you need to adapt your statistical threshold. Otherwise, you face the risk of inflating your false positive rate. And also, practically, it poses the question of when do you stop, actually? Because if you keep on collecting 1,000 subjects, you will eventually get a significant effect. There's always a difference, even a tiny one. So in sum, these questionable research practices inflate effect size, increase false positive rate, and poor reproducibility, and make you have poor reproducibility. Okay, and I'm going to finish with this. Actually, John Oliver, an American humorist, fairly famous, has a late show on TV, has a very nice piece on p hacking, quite unexpected from a humorist. He's done his research, and this episode is fun and very accurate on the topic. It was issued like a couple of years ago already, right at the peak of this the replication crisis that I'm sure many of you have heard about, and I'm not telling you what to do. Okay. Okay, so way too much data here. And no, really, I need to quit because we are really behind schedule, and I don't want to waste more of everybody's time. Okay, and I see the next speaker is waiting, and voila. Let me see, how can I get to the end of this presentation? I had a lot of things to tell you about pre-registration as a tool to overcome these questionable research practice. I thank you for your attention, and yeah, apologize for the glitches.